Thanks, Christoph. Yeah, today we're going to do some live experiment, but first I will just give a little introduction into Animal EEG. And yeah, we're gonna record a horse today live, so stay tuned. But first of all, I would like to introduce a bit the measurements we did with horses and dogs, because yeah, we just had access here at the company. So on the right side, you can see Santi, the company dog, and on the left side, you can see Dreamy, the not company horse, but from a working colleague, Susie. So generally, what we try to do is measure EEG with systems that we use in humans, so wireless systems, also for animals. And of course, one of the biggest limitations is here a standardized cap and electrode setup. So yeah, we try to build some prototypes, especially here in this case for Santi. We chose the medium-size dog skull, which would be E on the figure on the right side. And we used that to build our electro configuration with our EEG system. And here you can see a little overview of different positions and studies that used these electro positions with animals, with dogs in this case. So for us, very important was also, of course, the reference and grounding, because if we would use the ear or the lower back, like it's often the case, we would maybe induce some artifacts. So we tried to reduce that. So you will see in the next slides where we place them. This is also a very standardized setup with low amount of channels. We reduced it even a bit more, and then found the positions as you can see here. So we have FZ, CZ, P3, and P4 that we mounted with these bands onto the dog. Here it's just a very simple prototype that we did at the very beginning. And referencing ground we put here on the nose area, so we have less movement. And yeah, in the next slides you can see some prototypes that we built. So we put here referencing ground on the nose side, and we have these 4 electrodes, which are, as I said, FZ, CZ, P3, and P4, as in this standard setup that is shown in literature. You can also see here on the through the skull the approximated location of the brain of the dog. Of course, it's here also very important that we find the right positions to cover really the area of the brain, and we don't cover it somewhere where we don't really get the EEG. So these are some prototypes that we did, and this is Santi with the system actually. And I will just start this video. I hope you can see it, and that's how we mounted the electrodes on the back side here, P3 and P4, and on the front. And we had here the amplifier to transmit it. We did a couple of tests, and the EEG looks quite nice. And yeah, for now, with the dog EEG, we will do some experiments and trials, and with auditory vocal potentials, and also the same with horses. And shortly I want to mention that we tried different of the electrode systems. But in general, we use the dry hybrid black, so these electrodes, where we can also use gel. Maybe an application area of these systems, like is shown in literature by this study that was just recently published, that shows very nice mismatch negativity from objects that have been shown to the dog in this case, and they were named with the right name, and objects that were named with the wrong name. And this mismatch like phenomenon is also known in humans very much, which then also could represent that it matches the mental representation of the dog. So it's not just hearing this word, it really has a mental representation of the objects in the memory, which is showcased with one of the recent study. And you can see here the EEG system that was used is very much nowadays still in a very should be in a very quiet environment and also cabled. So we try to overcome that with our wireless system. And this is also one of the big reasons why we test the system now, to have yeah, a better usability with the animals, so they're not restricted to laying down or be sedated. And the next topic, also the special topic of today, is horse EEG. So here you can see the location of the brain of the horse, very much close here to the ears. You will also see then when we put our system where we locate it. And yeah, also we have here a big variance in head sizes. So we built some prototypes for pony and also for standard big size horses. And yeah, actually talking about the horse, I hear the horse already coming, so I can maybe show you quickly in the camera then with me. So if I proceed, also here is a big topic is the standardization of these electrode layouts. So there are a couple of layouts out there, and we choose for our prototype, as we used 4 channels again, the number A. So we placed 4 channels approximately in this area, and the reference and ground behind the ear for our measurements. So yeah, here you can see also how these straps are used to mount the headset. Maybe Susie, if you can come with the horse shortly, I can already show it in the camera, just that people believe that we have a horse here. So here is our subject for today, that's Dreamy. He's very interested in the camera right now. So, but I'll just finish the presentation and we do the measurement. So yeah, here you can see, for example, the setup of the pony that we also measured with these electrodes placed, the locations as mentioned. And on the right bottom, you can see it in the first 4 channels, the EEG of the horses, which has a very high amplitude also, especially because the school is here not as thick as with humans. So we even have a better signal. And if I proceed, I can also show you, again, the signal quality here looks very good, the power spectrum. And here again, how we mounted, maybe I will just start the videos. Here is the pony. So we have the reference. We used some gel for these test measurements and 4 electrodes on the forehead. And here the bigger horse, we fill the gel inside these hybrid electrodes. We can use them dry and with gel. And the reference and ground is here behind the ear. We have these rubber bands that we use to mount the electrode locations, and the amplifiers here on the back side mounted. With the new prototype that you will see today, it is set up a bit different. Of course, we did also a couple of different tests. In this case, we showed them delicious food and waited a bit, but of course, in the end, we feed them also with the good food, and we showed how the EEG behaves. And then also we have some tests with, yeah, in the inside. So you can see here, similar. And yeah, with that, I can already start the mounting of the displays or the live videos. Here you can see some videos from some tests, but I guess we could just do it in live. So if Susie comes to me now, and then Marco can share his screen now. So I will just stop and Marco will share from his screen. Okay, I hope you can see Marco. Wait a second, let me just mute and then you can speak with your screen, okay? Marco is connected.
Okay, hello to everyone. I am Marco. I'm from Naples University Federico II, and I'm working with Leo on this animal EEG acquisition and processing or studying. Now we are going to show you how we developed our cap. It has to be as less bulky as possible. And as you can maybe see from my camera, okay? Okay, you can see how the electrodes are placed. The electrodes are the hybrid ones. This is our device for EEG acquisition. And then, okay, and Then there is the reference that is placed something like 5 centimeters away from the left ear of the horse. Okay, now we can go with putting it on him, thanks to the help of Susie.
I have to put the hair a bit away, so
Yes, it's
exactly then
possible to look at this.
take care the reference has some contact. And we just mount them here. Put them and adjust them. Yes. Now we have a detailed look on the positions. Of course, we take care that we don't annoy the horse too much. So that's how the mounting basically works. We have these 4 channels with the basic station here.
Electrode positions. Here we got the amplifier, and then on the heart we have the reference electrode left here.
I hope you can see now better the video.
Okay, we moved a little bit the hairs so that it has better contact with the skull. Putting our amplifier on and starting putting some gel on it.
Make sure that the reference is touching the skin here. Yeah. So we adjusted a bit more.
Yep. You can see at the horse moment. Overall electrodes. It's really strict to the skull of the horse. That's the configuration. It doesn't hurt at all. The horse is really fine with that. Not restricted too much.
Okay, so I will share my screen. Maybe you can mute again, Marco.
Yeah.
And just keep on showing the positioning and check a bit the reference. So I will share again my screen right now with the model that we prepared and the experiment that we want to show. So basically we want to show some standard EEG and also some evoked potential experiments. So we have here our Simulink model with the amplifier in the input and the paradigm presenter. So what we try to do is now to run the system and check if we can see some data here. So try to do is record some standard EEG from these 4 channels and then also these EPs online. So we can check. So we have quite a good signal on 2 channels, but on 2 channels we need to adapt a bit more. I will just go now to Marco and help him with that. I hope you can hear the beeping already. So this is actually what we are gonna show so what we're gonna present these auditory evoked potentials to the horse, and we're gonna record this data and also show you the online EPs. So I'll just stop the presentation again. And yeah, so maybe Marco, you can turn on, then I'll adjust a bit. You can turn on
Okay. I already adjusted
I can
a little bit.
So I will have to check that the reference has good contact here. And also in general, that the system holds nice. It's okay. Maybe you can mute to the the electrode.
Okay.
This can explain a bit more the mounting, maybe, Marco.
Yes, the mounting is just as said before, we're just trying to don't hurt the horse at all, but at the same time placing the electrodes in positions of the skull that able us to record some really good EEG data. Now we will also look at them, so that will be better maybe. And yeah, that's everything that you have to know. Is just the position of the reference. This choose maybe. Still not that.
And I will add some more gel.
Mm-hmm. Yep. Sorry. Sorry, Dreamy.
Yeah, a bit of gel. And the horse gets, of course, a nice carrot.
As a reward.
The hair of the way.
Mm-hmm.
So it lets it.
Okay, what?
Okay, so the first two channels look quite okay. We have here the EEG of the horse. Of course, it's moving here also, as you can see in the background, but it looks quite good. And now I will try to open also here both channels for the EPs. So maybe you can unmute again, Marco. So mute again and mute, and I can unmute it. So I will run now the experiment again, and I will show you the EP scope also here. I hope you can see also the video of Marco. I'll just start here the experiment and open the vector scope. I don't know if you can hear the beeping, maybe not too strong. Maybe you can come a bit closer, Susie, with the horse. So the horse can also have this audio signal. Exactly, perfect. Can you hear the beeping? Is it okay? So,
Be silent for a moment, and we
should be a
can
bit.
check if we hear it. No, we don't hear it.
Okay. So here is the EEG scope. We can see the EEG of the horse. And if okay, so I will stop now. That's the paradigm. And I heard that you cannot really hear the beeping,
No, we are hearing
unfortunately.
before.
But yeah, we did an auditory stimulation here. We also tried to keep quiet. You've seen the EEG on the scope. So basically what we try to do is now a couple of different experiments, also visual experiments. And as you've seen, the setup was quite quick and we had a very strong amplitude because exactly what I said before of the distance to the school. Can you unmute again? Then we can go. Is it unmuted?
Okay, yes.
Okay, cool. So maybe I can shortly explain again the cap as it works. So what we did now is taking the amplifier here off and just plugging it on, and then it turns on and we connect with Bluetooth to it. And I think we had the electrodes a bit here covered by the hair, but they actually have a good contact. That's very important when you need to check the contact. So what we built here is basically a prototype. It's just some rubber bands where the electrodes are mounted, which you can do now for every size of the horse. So it's not fixed to this head shape. So and here also we built an adjustable strap, of course, so we can make it a bit bigger and smaller depending on the head. Most important, it also goes by the ear in front and on the back, so we can nicely and fast mount it. So you can see also, maybe you can walk around a bit with the horse now. You can see how this works when we walk around. Maybe you can talk, Marco, while you walk a bit, Pussy.
Okay, that's our experiment. The horse is really fine with it. It just doesn't feel it, basically. It's normal walk, and as you can see, this is acquisition and going to. So it is.
Yeah, exactly. So we'll show here again also the EEG while walking with the horse. It's very nice outside here. We had a really bad rainy day in the morning, but now it's perfect. So we were also a bit lucky. So let me just open again the device, and maybe Marco, you can just take off the amplifier and put it on again just shortly. You can show it also in detail. So Marco is unplugging with the magnet. Maybe you can, with one hand, you can hold it, right? And then you can just plug it on again. Mounted right, that's nice. So I can check again for the amplifier. What we also could use here, of course, would be the Oscar, so the artifact removal with movements, which would be very interesting in this case. Horse is happy. Is the horse happy, Susie? Mention, is it good? Yeah, it's good. You can talk to Marco. Yeah, so as I mentioned before shortly, what's also interesting is here using the artifact removal because of the movements, but basically the fast setup time is very important, and also that the animals can freely move, because that's often a very limiting factor for animal studies. So with a system like this that's just mounted to some straps with these electrodes, we could overcome these limitations and also then use it for research, which it would be a very good, yeah, benefit. We also thought about so I will just share the screen again, and then you should see the EEG again. Exactly, here we have it. Here we have it, and then maybe, I hope you can see it now. There is also this presenter window. So maybe you can walk around a bit with the horse again, Susie. You can walk a bit. Then you will also see that it's, well, at some point, of course, we will lose Bluetooth connection, and you should come back, because we have here a distance of, let's say, 20 meters now from my computer. So we are quite far, but still it records nicely. And yeah, so maybe you can come here, Susie, with the horse closer to me, and then stop walking, so we can see that it's exactly. So now they're really close and quiet, so you can see also the EEG got more stable. Now Marco walks by, and so maybe we can go give the horse some treats again. A carrot, we have a lot here. For the auditory experiment, we will have to make also a quiet environment, so the horse can focus also here on the beeps to see the EPs in this case. Oop. Yeah, so that's basically a very simple experiment that we ran here, like just putting on the cap and showing some EEG channels with the device that we have. And we tried also to record dry. We saw also some EEG. We just used some gel for better signal quality in this case. So maybe.
So Leo,
The horse is
do you
hungry.
do you see the artifact when the horse is eating?
Yeah. Can you hear me? You want to make the screen bigger or what?
I'm asking if you can see artifacts when
Okay,
the
sorry, Christoph, I
horse
didn't
eats
hear you
a...
because I was not on the phone. Now, Christoph, I can hear you.
No worries. So if the horse eats the carrot, do you see artifacts in the EEG?
Yeah, we see some movement, but actually not so much, because maybe you can see now when Marco, you can see the video. Even though there is the biting, the electrodes in the front are quite stable. Not as with humans, there are actually more movements. So if you go up and show the electrodes in the front, we didn't see so many artifacts as when humans bite, for example, which was very interesting, because maybe it's also connected because there are less muscles here. Yeah.
What about eye movements?
Yeah, that's also like these low frequency components we also saw, but we cannot really instruct the horse to perform, so we can't just feed them some carrots. So it's hard.
What about ear movements?
Yeah, exactly. So that could be a problem, ear movements. That's why we placed also the electrodes on the neck, on the back side, and not on the ear itself, because horses also, maybe Susie can tell me, but also like they show also with the ear movements a bit their reaction, of course, and where they're focusing to, and maybe even some emotional indicator. I don't know especially, but maybe Susie says yes, so I assume that's the case. Also with dogs, that's of course especially tricky. So we want to, yeah, we could not mount here the reference on the ears.
And could you ask Susie maybe to shade the eyes, so to simulate to close the eyes? So we cannot really ask the horse to close the eyes, but maybe Susie
Yeah.
can. I don't know if her horse likes that, but maybe we see some alpha coming.
Yeah, we can give that a try. Susie, do you think the horse is okay when you shade the eyes a bit, so it's like closing the eyes?
So that's horse alpha.
Oh yeah, that
So-
looks like it works. So I'll just run the system again, okay, and then we give this a try. That's a good one.
So has anybody seen horse alpha so far? Let me know in the chat. Nobody. So let's do it.
Okay, then we do that first time. So also maybe important to mention, because alpha, yeah, we know that is especially prominent in the occipital region of humans. We have definitely here, like, of course, another distribution of these functional areas. Actually, here on the back side, it would be on the back side of the strap. So we are more in the motor area, actually, that we record with our sensors. Yeah, maybe somebody that does here animal research can tell me that better, but I think here is also reduced the prefrontal cortex. So we have more on the frontal, the motor, and on the backside, the visual part of the brain. So let me just.
So an expert is suggesting that you should all sing Italian songs because this is relaxing for the horse.
Okay, that's a good, very nice expert suggestion. So we should all sing now, as we have 3 Italian guys here. I guess that shouldn't be any problem. So yeah, I don't know, Susie, is there something that, I mean, Susie can do that. We will try that now. Maybe some sounds also that the horse likes. You can make them simple, very simple, like some clicking. How are the sounds, Susie, so we can hear it? Like this. No, what is the? Yeah, you can do it. I don't know if you can hear it. You can hear these specific sounds that Susie makes, because yeah, it's just to get them for attention. Yeah, Marco will turn on the audio, and then you can listen to the sounds.
Okay, please, Susie, do it.
Horse is reacting very much. So
So Martin is suggesting the Italians should sing Adriano Celentano Azzurro.
so guys, now we can share this again. Maybe I can talk on my. Okay, Marco, turn off his. So just reopen that. Yes, so window.
So Leo, could you also maybe show us the EEG data stream while you're doing
Yeah, I'm
some
just
slide?
doing that now. Sorry, I was just trying to share my screen. So we have here again the first 2 channels, and maybe Susie, you can try to cover the eyes now of the horse, so we can really try that. Maybe we can see. I'm not quite sure. We never tried that now. A bit more. So you see here some artifacts of the people touching around in the amplifier and the electrode.
Susie's jumping. Seems
So
like the
now
horse
they're
doesn't like it.
closed. I don't think so. Maybe.
Here was a short alpha burst.
Yeah, we can do the FFT. Let's see if it's working.
But the horse doesn't really like it, so no worries.
I don't think so. That's why we have also some movements here. Yeah, so we can leave it now, Susie. You don't have to. Now it's happy
So
again.
this is all a hint that you should actually sing.
Okay, yeah, it's a hint that you should sing, guys. They're not very happy about that, Christoph, but yeah. Yeah, this would also be a nice auditory experiment for all of us. No, but it's yeah, the EEG is quite nice, even though when moving around with the horse. But yeah, of course, the more quiet, the more stable the environment is.
How are you filtering at the moment the EEG? Now we have some alpha, you see the bursts. So what are the bandpass filter coefficients you're using?
It's 2 to 30 in this case, just to display it a bit nicer. Exactly.
And if you look at the spectrogram of a horse, so is it comparable with humans, or they have more alpha, theta, delta? Do you know?
Well, actually, I don't quite know. Which we saw last time we measured a bit more activation in the higher, so in the power spectrum we had a bit higher frequencies actually. But yeah, that's something we definitely also.
So if I see the EEG, I would say that's EEG from Leo.
Now, because it's so much alpha, because
yeah, you need more work. Yeah,
exactly. That's why. Yeah. So yeah,
what's the name of the horse? Somebody's asking.
Dreamy.
Dreamy.
Dreamy is the horse,
Yeah,
and we have also
so much alpha.
Yeah, it's more even lower. What's the name of the pony? Merlin and Maxi King is also the name of another horse. So we have access to 3 horses here. So we have a lot to measure. Maybe I can show again a bit some slides from the recording we did today.
Leo, it would be super nice. I don't know if it's working and you can show it, but to see the raw data while you are doing the beep, because if there's a strong auditory evoke potential, we should actually be able to see it in the raw EEG.
Yeah, I will try it now. So I could just stop and try the recording again with the auditory. I don't know if you could hear the beep actually, because with sharing the sound, it's not so easy.
A little bit from the cell phone afterwards.
Sorry, that's like easy to share.
But some people are wondering, of course, while somebody would record EEG data from a horse, but animals have, of course, very interesting sensory systems, and it's worth to study them. So dolphins, for example, have a very good auditory system. Tomorrow there will be a lecture about EEG recordings in dolphins, and dogs have very good smell, and so on. And that's a fascinating research area. So you can find out more about sensory processing.
Can you come a bit close? Exactly. So now we have the beep.
You give me the audio. If you give me the audio,
yes, I will give you the audio. Now we had some movements.
My problem is that we don't really hear the beep.
There's a 250.
It's okay, Leo. We cannot hear the beep, so it's difficult to localize the auditory evoke potential, but it's fine.
Yeah. Beeping sound. Yeah. So
I close my microphone.
Exactly. So that's also something we try to do now with dogs and also other horses. Basically, the mounting of the devices is very simple. So as you can see now here, it's very easy attached. And yeah, with some gel, you get really nice signals.
So Sloppy is actually saying Zoom is suppressing the beeps because it's considered as noise, and then it's suppressed. That's the reason why we don't hear it.
Yeah. Yeah, we thought about also making some visual evoke potentials, but yeah, for that it's well, not so easy to mount displays and how to present it. I guess we would need a big display to show, and here at the farm it's not so easy. But I guess there are also even head-mounted display for horses. I saw something out there online. This would be very interesting to investigate, actually. Yeah, so this was a short insight into our animal EEG. Is Dreamy still happy or already? Yeah, okay, so Dreamy is still happy. So I guess we'll proceed with maybe an experiment. Are there any more questions for now, Krista?
There are some questions. We can do it now, but you could also. Do you have another experiment with Dreamy, or you want to show some slides?
Basically, I can show some slides because that's from the tests we did before also.
Then maybe we do the questions first and then we go to the slides. So Andre is asking, how do you plan to use the EEG for horses in future?
So basically, we're in contact also with some institute and people that work with animals here. And we are, of course, hoping for input from their side because they're very experienced. But there is a lot of research out there, of course, with human-animal interaction. For example, horses are used in people therapy. So in therapy with humans, which of course is an interesting topic. And there are also studies out there that investigate like hyperscanning wise EEG from humans and horses while riding the horse. I can also send you the link for that. So this would be a very interesting topic because with the device now, for example, we could just easily put it on a human and also on the horse. So we would have, yeah.
Brian is asking if there are any studies on how horses respond to music.
I don't know that, but yeah, this would be also very interesting.
That's another hint for Azzurro, yeah. That's a funny one. Could you extract with the brain-computer interface the voice of the horse?
Well, that sounds very interesting. Communication-wise, it would be very interesting, but if we can do that, that is to be found out. Yeah.
So now Dreamy would say, leave me alone.
Yeah, she already looks like that.
Elida is asking, how many channels are you recording?
In total, we record 4 here. So we have reference ground on the neck behind the ear. And the 4 channels that you can see here in the center is the amplifier, the wireless amplifier, and top right here exactly you can see the 4 channels. So these are the 4 clips that are plugged onto this this hybrid electrode.
These are for the moment all the questions. You, Leo,
okay.
Please
yeah.
proceed to the slides. Thank you very much
Then
for the demo, Susie. Thanks for Dreamy. Very nice. And while Leo is showing the slides, you can, test a little bit the singing in the background so that you can do it at the end. Let Marco and the Italians know.
Okay, Christoph. We will do that. So, well, in the slides, I just wanted to show you some videos of today that we did before when testing. So for mounting the headset, we performed it like that as you've seen before live, but here maybe a bit more in detail. So we just have here these 2 straps. We had to remove the hair, and that's basically it. So that's basically it. Exactly. Also, one idea we had was mounting it on the leather holsters, on the leather straps directly, but with this, it's just way easier to adjust. And then you can see here when we just put on the amplifier, we just plug it on with magnets, can be plugged on, and then it's ready to measure. In the next step, here we show again how it looks when the horse comes along, and we pop on the amplifier, and then it's ready to measure. Also outdoor, as I said, it's Bluetooth, so we would need a distance of, let's say, 10, 20 meters, but it also can happen with a bit more distance, probably needs to be found out. And then what we do is add some gel. So also with the horse, this worked pretty nice. Even though Susie told me that, because I thought, yeah, horses probably don't feel that so sensitive. Susie meant even on the opposite, they're very sensitive. So yeah, means also they were quite okay, and they can wear the electrodes and are not yeah, oversensitive with it. Even though Susie told me they're very reactive to any touching. And then when you have applied it, again, here the application procedure, we can also record. So today we recorded this EEG with the horse. We had it as now placed, and we could record. Even though with a very high amplitude compared to human EEG, 150 micro, even a bit more than with humans, because also of the fact that the school is here not as thick, probably. So yeah, that's what we tried today, and we also tried to run some more experiments now. So yeah, that's it from my side actually. Christoph, we're a bit early, but we also started a bit earlier. So we discussed something specifically about the dog EEG.
Are there any questions for Leo and Marco and all the others in the meantime? Otherwise, thank you very much. Make sure to make a group picture of you guys with the horse, so that we remember it in 10 years. First horse recording at Spring School. And hopefully it inspires a lot of more research in this domain. Thank you very much, guys, and see you back in the office afterwards.
Bye, see you.
Leave the horse there.
Oh, I wanted to bring it.